Welcome to our YouTube channel of Applied Forensic Research Sciences. My name is Shivani Ghodke and I am the intern in AFRS. Today's topic of presentation is how saliva can be identified. And here we will discuss the introductory part, composition of saliva, and preliminary and confirmatory forensic test for the identifying saliva sample and its significance. On an average, saliva secretes 1 to 1.5 liter of saliva every day, and saliva is a fluid which is composed of water with the electrolytes and enzymes, and it is secreted by parotid and submandibular salivary glands, and it is also seen in sexual assault cases. Saliva is very important corroborative evidence. Saliva sample can be identified or may be analyzed from various types of surfaces from crime scene such as body parts, paper, envelope, cigarette butt, plastic, glass bottle and metal cans and so many. And saliva secretes the enzyme amylase which can break down the polysaccharide into the monosaccharide. Let's say example like starch which can be break down into glucose and maltose. Starch contain two types of glucose monomers, that is amylose and amylopectin. Let's see the composition of saliva. Saliva is made up of water and solids. It contains 99.5% of water and 0.5% of solids. And in solids there are organic substances, inorganic substances and gases may be present. Saliva is formed by three pairs of major salivary glands, that is parotid, submandibular and sublingual gland. And mostly the digestion of carbohydrate in the diet begins with the oral cavity, where amylase is a saliva breakdown and carbohydrate such as starch. And detecting the amylase enzyme which indicate the presence or absence of saliva. Let's discuss the presumptive assay. It is simply the preliminary assay for the saliva identification and mostly used to detect whether this given sample contains saliva. And these tests are quick, inexpensive and provide the first indication of presence of saliva. Visual examination. When searching for biological stains like semen and saliva, lightning techniques can be used to make them visible, and one of the technique is fluorescence, which involves specific wavelengths of light to make the stain glow. And saliva stains like 470 nanometers excitation wavelength can be used with the orange goggles to enhance the fluorescence. However, the fluorescence of saliva stains is usually less intense than that of seminal stain because semen contain more fluorescent compound than saliva. And the same lightning techniques can be still used to search both type of stains. Microscopic examination can be done with the histological staining like Giemsa stain is used, and this is performed to identify the buccal epithelial cells which indicates the presence of saliva stain. And here we can see that there is cell membrane, nucleus may be present and cystole is present. Mostly performed test for the identification of saliva is starch iodine test, and the presence of starch is identified using iodine, and this test can be performed by in test tube and in agarose gel. Let's see the test tube method. Take two test tube, one with the saliva and other with the without saliva sample. Add 2-3 drops of soluble starch solution in each tube and mix it and incubate it tubes for the 1 hour at 37 degrees Celsius. And after some time add 2-3 drops of lugol iodine reagent and observe the color. In one test tube there is no color, which is due to the starch is first hydrolyzed with the amylase and then mixed with the iodine solution, and blue color is not obtained because the starch has been broken down into glucose and maltose. And in other test tube there is only starch and iodine present, which can be mixed with each other, and it form the blue color. And this is the indication of absence of saliva. And when it get transparent solution, it indicates that the saliva is present in that sample. And basically it is not the confirmatory test for saliva, it is only used for the preliminary test. Same test can be done with agarose gel. Make agarose gel in petri dish, make the sample wells and load the question sample in the center of the petri dish and keep it incubate for the overnight. Next day, add the Lugol's iodine solution in it. We will observe the blue color to start a starch containing gel. And here amylase is present in the sample from saliva. It diffuses from the sample well and breaks down the gel starch as it does so, and the sample well clear perimeter denotes the presence of amylase. Here we can see the two petri dish. One is in one petri dish there is saliva present and there is a clear perimeter is formed, and other test tube there is absence of saliva, so there is no clear region is formed. This can be also called as the radial diffusion assay. Here we can see that we are loading the sample, obtain the clear area, and graph will be plotted like amount of amylase versus the size of clear area. And here the size of clear area is directly proportional to the amount of amylase in the sample, and we can determine it and measure the diameter. Next is calorimetric assay. Here we will see the AFRS reagent we use to identify the saliva sample. Starch is basically linked to the colored dye, and the presence of amylase releases the dye. And there are two methods we can use using AFRS reagent, like there is press test and tube test. Let's discuss the first of all tube test. A small amount of piece containing stain is placed in the test tube, and water and AFRS tablets are added in the test tube. Then we will heat for a few seconds and centrifuge to pellet tablets, remnants and piece of clothes. And amount of color in the top of the liquid is measured by the spectrometer. And press test. AFRS reagent spread on the filter paper, then allowed to dry the AFRS paper placed on the area to be tested. Paper is sprayed with water and pressed against the area. Paper is left on the area and watched for blue color changes, and this can be watched for up to 40 minutes for the change in color. Here we can see in the picture. There is another presumptive test like calorimetric assay. We can identify using the Saliva reagent. Saliva reagent kit in available. Another commercial available calorimetric assay and has been validated for the saliva identification. And its manufacturer also produces the spray kit which can be used for amylase mapping. Then we will discuss the confirmatory assay. Here immunochromatographic assay is used for the identification of saliva. Also the ELISA is used. In immunochromatographic assay, there is commercially developed kits are available for the testing of the sample. Here we can see in the picture. And this method is based on antigen-antibody interaction principle. One such kit is also known as the RSID saliva kit, and It indicates the presence of saliva, and it can be ascertained by the appearance of pink line in the saliva kit, and in taste zone and saliva control zone we can observe the lines, and this technique can be detect saliva as low as the 1 ml. There is another test like RNA based test. Basically, mouth contain these cells that are specific to it, and these cells contain specific protein which encode the genes that make them identified. And there are two genes like HTN3 and SHAT. Basically, these are two genes which encode the special protein, and HTN3 genes encodes histamine 3 protein which is involved in defense mechanism in oral cavity, and SHAT encodes slytherin which is prevents the precipitation of calcium phosphate salts in salivary glands. And here we will discuss the whole test for the identification of saliva. So what is the conclusion of this PPT presentations? Like in saliva is very valuable biological fluid in forensic investigation due to its abundance, and easily collection is done, and the potential for DNA analysis, and this preliminary and confirmatory test can be used to identify the presence of saliva sample in the crime scene, and DNA analysis further can be provided the positive identification of source of saliva, which can be used for the species identification, and saliva can be identified the source of biological sample, and also on various cases it provide digital DNA evidence, and bodily fluids are not present. And lastly, the forensic significance of saliva. Saliva found at crime scene and mostly used to identify the perpetrator of the crime in the cases of sexual assault, and saliva can be used to exclude the suspects in the investigation. If DNA analysis of saliva from the crime scene does not match the DNA of the suspect, then they can be excluded from the investigation, and it is used to identify the drugs or other substances. Here you can see the references. Thank you everyone for listening to this topic. I hope you learned something productive from this. If you have any doubts, kindly comments down. Please like, share our video. Also subscribe to our YouTube channel.